Hello students, let us start with the next chapter of biology, that is body fluids and circulation. So first of all, let us try and understand what is circulation. Circulation can be defined as the transport of nutrients, oxygen, carbon dioxide and excretory product to the concerned tissues or organs. It is basically the movement of substances to different parts. So it will be different in different organisms. The mode of circulation will vary. For simple organisms, they will use diffusion because their body is very simple. When the complex organisms have to circulate body fluids, then they will use a different method of circulation. Let us look at these systems. So there are various types of circulation. One is open and the other one is closed. An open circulatory system is where the blood is pumped by the heart and it passes through large vessels into open spaces or cavities called sinuses. We can see this circulation in arthropods or mollusks. Then let us understand what is closed circulation. So a closed circulatory system is where blood pumped by the heart is circulated through blood vessels. It is more advantageous as the flow of fluid can be very well regulated. It can be very precisely regulated. We can see this circulation in annelids and chordates. All of the vertebrates have a muscular chambered heart. Now, how many chambers are there? That will differ in various organisms. Like fishes have two chambered heart with an atrium and a ventricle. Amphibians have a three chambered heart with two atria and a ventricle. Reptiles have a three chambered heart with an incomplete partition for the ventricle. Crocodiles have a four chambered heart, and this is how it varies between different organisms. The circulatory pathways are also different for different organisms. As we discussed earlier, the patterns of circulation in vertebrates can be single, incomplete, or double circulation. Let us look at the single circulation. Here, it is mostly seen in fishes. The heart receives impure blood and it is purified. So you can see here the blood is circulated only once. So the deoxygenated blood will come to the heart. The blue colored is deoxygenated. It goes to the gills, which is the main organ where diffusion happens. It is converted into oxygenated blood. As CO2 is out, oxygen is in now. And then this oxygenated blood is sent to all the body parts. Again, the body parts use it, so it is converted into deoxygenated blood, which is sent back to heart, and this way the cycle goes on. We see incomplete double circulation also in amphibians and reptiles. So here what happens is the left atrium, that is the top chamber, it receives the oxygenated blood from gills, lungs or skin, and then the right atrium gets deoxygenated blood from other parts of the body. However, here there is mixing of blood, right? So the ventricles are now pumping out this mixed blood everywhere. Then we see double circulation in birds and mammals. We humans, we have this double circulation. So here the right atrium receives the deoxygenated blood. It passes it into the right ventricle. Then the left atrium receives the oxygenated blood, which it passes again to the left ventricle. And the ventricles pump it out separately without mixing up. So this is one important feature where the oxygenated blood and the deoxygenated blood are not mixing up together. If we look at the human circulatory system, then it consists of following things. First is the blood vascular system and the lymphatic system. The lymphatic system will have lymph vessels and the lymph node. The blood vascular system will have the heart, which pumps the blood around the body. It has blood which is made of liquid part and the solid part. The liquid component is the plasma. The solid components are RBC, WBC and platelets. The WBCs can again be divided into granulocytes and agranulocytes. Granulocytes means they have granules. So these can be divided into neutrophil, eosinophils or basophils. The agranulocytes are lymphocytes and monocytes. So lymphocytes, we can divide them into B lymphocytes and T lymphocytes. The blood vessels are arteries, veins and capillaries. This is how the circulatory system is formed. Now let us study more about blood. So the blood, as we saw earlier, is made up of plasma, that is the liquid part, and the solid components. So the solid components are the cells, WBC, RBC and platelets, and plasma comprises of 55% of the blood, which is the liquid part of the blood. Okay, it is straw colored and it is slightly alkaline. Slightly alkaline means it has basic pH of around 7.4. So let us look at the functions of plasma. Plasma is responsible for clotting. Whenever we get a cut, then we observe that after some time our blood stops coming out. So that is called clotting. Okay. When the technicians, the lab technicians collect our blood, then they remove the clotting factor. So when they remove the clotting factors, we call it as serum. So the plasma consists of water, which acts as a solvent. Then it has plasma proteins, fibrinogens, globulins, and albumins, which is responsible for blood coagulations. The globulins play an important role as an antibody, which helps us to defend. Albumins help to maintain the osmotic balance and they regulate the blood pressure. Glucose, amino acids, lipids, cholesterol are also present in the plasma, which are required for energy production and growth. Then it has inorganic ions like sodium, magnesium, chlorine, HCO3 and calcium, which help in maintaining the osmotic balance. Then it has various gases, carbon dioxide, oxygen, nitrogen, which are essential for transport. Now let us look at the solid components. So as we saw earlier, there are three components: RBC, WBC and platelets. RBC, as the name suggests, is red in color. So why is it red? It is red because of the hemoglobin present in it. There is about 12 to 16 grams of hemoglobin per 100 ml. RBC has a very special shape. It has a biconcave shape. It is one of the only cells without any nucleus. So this is a special feature. It does not have nucleus, mitochondria or Golgi bodies. Its function is to transport oxygen and CO2 throughout the body. Its lifespan is about 120 days. There is about 5 to 5.5 million of RBCs per mm cube. Then we study about WBCs. So WBCs are generally colorless. Their lifespan is about 1 to 15 days. We see 6,000 to 8,000 per mm cube of WBCs. They are found generally in bone marrow, lymph glands and spleen. The most important functions of WBCs is that they help in the immune system. They help us to be immune to various diseases by doing phagocytosis, by producing various antibodies. They can be nucleated. On the basis of that, they are divided into different types. So these types are neutrophils, eosinophils and basophils. All of these neutrophils, eosinophils and basophils are granulocytes. So neutrophils play an important role in phagocytosis. They are soldiers of our body. They engulf the things which are not required. Acidophils, they resist infection and cause allergic reaction. Basophils secrete histamine, serotonin, heparin and cause inflammatory reaction. Monocytes also play a role in phagocytosis. Lymphocytes are B lymphocytes and T lymphocytes. They play a major role in immune system of the body by secreting antibodies. Then let us look at platelets, which are also called as thrombocytes. These are generally colorless. Their lifespan is about 7 days. They are 1.5 to 3.5 lakhs per mm cube. They are formed in the bone marrow. They are megakaryocytes. They are non-nucleated cell fragments. And as I told you earlier, they play an important role in the blood clotting. So whenever we have a cut, they start blood coagulation. It is a mechanism for homeostasis. That is, we cannot afford to lose so much blood from the body. So they prevent the blood loss which can occur. So how does this blood coagulation occur? Whenever there is an injury in the body or damage, the signal is sent to the platelets and tissue. They will convert thromboplastin into prothrombin by this enzyme called thrombokinase. Then the prothrombin is converted into thrombin, which in turn converts fibrinogen into fibrin. Now fibrinogen is soluble whereas fibrin is insoluble. So because fibrin is insoluble, it forms a mesh-like plug which prevents the blood from going away. So in this way, clotting is completed and all the damage of the body is prevented. So this leads to homeostasis when an activated platelet stops the excess blood from the body from going out of the body. Now let us understand about blood grouping. So we might have heard many times about people talking about their blood group, how it is A positive, B positive. Let us try and understand this. So blood groups were first discovered by Karl Landsteiner. It includes the ABO grouping and the RH grouping. Now let us see what is this ABO grouping. It is based whether the antigen is present on the surface or not, right? So the plasma contains two types of antibodies, anti-A and anti-B. So if a person has antigen A, then he will have anti-B antibodies. If the person has antigen B, then the blood will have anti-A antibodies. If the person has both the antigens A and B, then neither anti-A nor anti-B will be present. If the person does not have antigen A or B, then both anti-A and anti-B antibodies will be present in the blood. Okay, so this is how we have different blood groups. Antigen on the surface, the antibody will be opposite. So if the person has antigen A, the antibody will be anti-B. If the person has antigen B, the antibody will always be anti-A. If the person has both the antigens, then the blood will not have any antibodies. So based on this, we can decide to whom the person can donate the blood. If the person gives the blood to a wrong blood group, then there might be disastrous reaction. So a person with blood group A can donate to A and AB and receive from people with blood group A and O. The person with blood group B can donate to B and AB blood group and can receive from B and O. A person with AB blood group can donate to OAB only and they can receive from A, AB and O. And a person with O blood group has anti-A as well as anti-B body. So these people they can donate the blood to everyone, A, B, AB as well as O. But they can receive blood from people of O blood group only. So this is how the blood grouping works. Generally, antigen A reacts with anti-A, antigen B will react with anti-B, and the mixing of bloods with interactive antigens and antibodies causes clumping of the cells, as we can see in the figure. So this is also called agglutination of RBCs. That's why while donating the blood, it is very important to check the blood group. So this is how we find out that people with O blood group are universal donor and AB group are generally universal recipients because they can accept from all blood group. The other method is RH grouping. So RH is basically rhesus. Rhesus factor is an antigen which is found on the top of RBC. So if this RH is present, then it is said to be Rh positive blood group, and if the Rh is not present, then it is said to be Rh negative blood group. Okay, so nearly 80% of humans are Rh positive. Now, anti-Rh antibodies are not naturally found in our body. So if a Rh negative person can receive the blood from Rh positive person only once, but it causes development of these anti-Rh antibodies in his blood. So a second transfusion of Rh positive blood causes something called as agglutination. Therefore, we have to match the Rh group before the donation of blood. So Rh positive person will be able to donate the blood to Rh positive person, but if he donates to Rh negative person, then we'll have a problem. Whereas Rh negative person will be able to donate to Rh positive as well as Rh negative. Now, this is especially significant in pregnant women. There is something called erythroblastosis fetalis. So here what happens is there is Rh incompatibility between the mother and the child. What happens is the Rh negative blood of a pregnant mother and the Rh positive blood of the fetus, they react. The mother is Rh negative, so the antibodies for the Rh is not present in the mother's body, whereas the fetus is Rh positive. So the mother's body essentially thinks that fetus is some foreign object. Okay, so the Rh antigens do not get mixed with the maternal blood in first pregnancy because the placenta separates them. But if there is mixing, then it will result in problems. But at the time of first delivery, there is a possibility of exposure of the mother's blood to small amount of Rh positive blood from the fetus. So this induces formation of Rh antibodies in the maternal blood also. So in case of a subsequent pregnancies, that is when she gets pregnant for the second or the third time, the Rh antibodies from the mother leak into the fetal blood, which is Rh positive, and they might destroy the fetal RBCs because they think it's some foreign object. So this is fatal to fetus or can cause severe anemia and jaundice to the babies. So this condition is called erythroblastosis fetalis. This can be avoided by administering anti-Rh antibodies to the mother immediately after the first delivery, so there is no risk when there is subsequent pregnancies. Now let us understand about different types of blood vessels. So there are three main types of blood vessels in the body. They are arteries, veins and capillaries. Arteries generally carry blood from heart to other tissues. They contain oxygenated blood. Their smaller branches are called arterioles. Then we have three layers of arteries. There's a inner layer, tunica intima, tunica media, which is the middle layer, and the outer layer, which is called tunica externa. So the inner layer is made up of squamous endothelium. The middle layer is made up of smooth muscles and elastic fibers. The outer layer is made up of fibrous connective tissue with collagen fibers. The veins carry blood towards the heart. They contain deoxygenated blood. All the veins contain deoxygenated blood. There is one exception, that is the pulmonary vein. The smaller branches are called venules, and the veins are also three-layered, but tunica media is comparatively thin over here. Now let us look at capillaries. So in tissues, arterioles divide into thin-walled and single-layered vessels. They are called capillaries, and they unite to form venules. Let us look at the structure of the heart now that we understand all the blood vessels. So heart is a mesodermally derived organ which is located in our chest on the left hand side. It is protected by a double layered pericardium. The pericardial space that is between the membranes, it is filled with something called pericardial fluid. This fluid helps in reducing friction between the heart walls and the surrounding tissue. If we look at the structure of the heart, then the heart consists of four chambers. There are two upper chambers and two lower chambers. The walls of the cardiac muscles of the ventricles are much thicker than that of the atria. The atria are separated by interatrial septum. So septum is basically what is dividing the heart into the four chambers, and it prevents mixing of the blood. The ventricles are separated by interventricular septum. In between atrium and ventricle, there is a thick fibrous atrioventricular septum with an opening, a tricuspid valve, that is, it has three flaps. They guard the opening of right atrium and the right ventricle, and a bicuspid valve guards the opening between left atrium and left ventricle. The tricuspid and the bicuspid valves allow the flow of the blood only in one direction. So it is like a door which closes so that oxygenated and deoxygenated blood do not get mixed together. The right ventricle has an opening to pulmonary artery and left ventricle has an opening to aorta. These openings are called semilunar valves. They prevent the backflow of the blood. If we look at the conductive system of the heart, then it consists of the nodal tissues, bundles, and fibers. The nodal tissues are specialized cardiac musculature which are present in the heart wall. They are of two types. We call them as SA node and AV node. SA node is the sinoatrial node which is present in the right upper corner of the right atrium, as you can see in the picture here, and the AV node is present in the lower left corner of the right atrium close to the atrioventricular septum. From this AV node, a bundle of fibers, atrioventricular bundle, passes through the atrioventricular septa and it divides into right and left branches. Each branch passes through the ventricular wall on its sides. In the ventricular wall, it breaks up into minute fibers. These are called Purkinje fibers. These fibers, along with the bundles, are called bundles of His. So the nodal tissues, they generate an action potential without any external stimuli. That is, they are auto-excitable. That's why our heart beats without any stimulation from outside. The SA node initiates and maintains the contraction of the heart by generating these action potential at about 70 to 75 per minute. So it is called natural pacemaker of the heart. I hope you all understood about the structure of heart in this video. In next video, we will learn about the cardiac cycle. Thank you.